Welcome to our poster on annotating the SARS-CoV-2 genome with conservation states. Our work is motivated by the fact that we urgently want to better understand the SARS-CoV-2 genome and its variations. Multi-strain sequence alignments, where various coronaviruses are aligned against SARS-CoV-2, can provide useful information in doing so. Cons-HMM was recently introduced as a comparative genomics method that systematically annotates a given genome with conservation states that reflect combinatorial and spatial patterns in a multi-species sequence alignment. As shown in this figure in the bottom left, we provide sequence alignment of coronaviruses as input to Cons-HMM, and as a result, we learned conservation states annotating the SARS-CoV-2 genome at a base pair resolution. Cons-HMM uses a multivariate hidden Markov model to model whether bases from non-reference sequences align to and match bases in the reference genome. Unlike previous comparative approaches, Cons-HMM does not explicitly use any phylogenetic information apart from the input alignment generated using phylogenetic trees, and therefore does not make any strict assumptions on the phylogenetic relationship among sequences. This allows Cons-HMM to be more flexible than methods that rely on phylogenetic information. In this work, we apply Cons-HMM to two multi-strain sequence alignments of coronaviruses, both of which have SARS-CoV-2 as their reference. The first one is the sarbecovirus alignment, which consists of SARS-CoV and 42 sarbecoviruses that infect bats. The second consists of 56 coronaviruses infecting various vertebrates ranging from human to birds. By applying Cons-HMM to these two alignments separately, we learn two sets of conservation states, 30 states from the sarbecovirus alignment and another 30 states from the vertebrate coronavirus alignment. Here in the center, we show the emission parameters learned by the multivariate HMM, each heat map corresponding to each alignment. In each heat map, each row corresponds to a state, and the left side shows the probability of a base in a non-reference strain aligning to SARS-CoV-2, where each column corresponds to a non-reference strain in the alignment. Similarly, the right side of the heat map shows the probability of a base in a non-reference strain matching or having the same nucleotide as SARS-CoV-2. For example, the top line of this heat map for the sarbecovirus model shows state S28, which has high align and match probabilities for bat coronavirus RATG13 only, which is the closest strain to SARS-CoV-2. The state, therefore, is highlighting bases unique to SARS-CoV-2 and its closest strain, bat coronavirus RATG13, and missing in other sarbecoviruses. These emission parameter heat maps overall demonstrate that the states capture distinct patterns in the input alignment, grouping bases that likely share a similar evolutionary history. We observe also that the states have distinct enrichment patterns for genes and regions of interest, often in agreement with previous work. For example, the state that we just discussed, state S28 from the sarbecovirus model, is strongly enriched for the human ACE2 binding motif, which is of strong interest in understanding how the virus infects human cells. And the state, as we saw before in the emission parameters, highlights bases unique to SARS-CoV-2 and bat coronavirus RATG13. We also observe that certain states are strongly enriched or depleted of mutations observed in SARS-CoV-2 sequences collected worldwide. Certain mutations previously highlighted as potentially consequential are found in states depleted of mutations in general, such as state S17 from the sarbecovirus model. This is consistent with the idea that bases under negative selection are likely to have strong consequence when mutated. To take advantage of this information, we generated genome-wide tracks based on statistically significant depletion of mutations observed in conservation states. Here we show an example of what the tracks look like in the genome browser, where bases annotated by states strongly depleted of mutations are scored high, and bases in states strongly enriched of mutations are scored low. When it comes to the spike protein's receptor binding domain, we see agreement between our tracks and experimentally measured changes in RBD expression due to mutation, which suggests that our track may be highlighting bases where novel mutations are more likely to be consequential. Overall, we expect these genome-wide tracks based on state depletion of mutations and the Cons-HMM conservation states to be a resource for understanding the SARS-CoV-2 genome and its mutations. More information on this work can be found in our publication, which is shown in the bottom right corner. Thank you.